me introduce you, and then we'll go ahead and get started. Our speaker today is Dr. Kaitlyn Sarlo. She's an animal scientist researching mastitis at USDA here in Ames. Her research focuses on understanding the role of genetics in mastitis susceptibility. We had alluded to the animals on their farm is from an original herd at Minnesota. And before I misstep, I will turn the podium over to Dr. Sarlo.
Thanks, Fred, and thank you all for sticking with me. I cannot tell you how much I appreciate it. These are nice slides that they come. So like Fred said, my name is Kaitlyn Sarlo Davila. I'm an animal scientist at the National Animal Disease Center in Ames, Iowa. For the most part, the National Animal Disease Center is a really, really cool place to work. The major drawback is all the security we have, both physical and IT. So we are actually co-housed with overall centers called the National Center for Animal Health. And so we have both the USDA Ag Research Service side, which is the National Animal Disease Center, where we do research on different diseases. And mastitis is probably the least spicy disease that we work on. We work on some things that have, implications for human health, like brucellosis and tuberculosis. And so we have some pretty serious physical security. We're co-housed with the Center for Veterinary Biologics, which is under the Animal Plant Health Inspection Service. And so if you can't tell, I pretty much live in alphabet soup. And so what they are responsible for is basically the go-ahead for any and all animal vaccines used in our country. So we have some pretty serious IT security because I don't unfortunately have any state secrets on my computer. But you can imagine if you're a company that's developed a vaccine and you're going to give that proprietary information to somebody to test, you're going to want to make sure nobody can get emissions. So again, sorry for that. So I have actually been a scientist over in the mastitis research project since April of this year. And we're actually funded through a congressional allocation. And so the name of our project is Genomic and Mitigation Strategies to Control Mastitis. And so I am a trained geneticist. I'm going to focus on the genomic aspect of that. And so again, it's a really cool place to work. And one of the reasons I really was excited to start this job is because my predecessor, there was a scientist named John Milpolis who did mastitis research for years, absolutely phenomenal man, unfortunately lost his battle with cancer in August of this year. So unfortunately, I'm the only scientist in mastitis right now. None of my research would be possible without the stuff he did. Okay, so if you just want to scroll down, this is where we are.
Right here.
Timing. Oh, one more up.
Nope.
Okay, yep. So there you see the National Animal Disease Center, very fancy building up top. I was just discussing the tremendous work of John Milpolis, without whom my current work would not be possible. So if you joined in the beginning, just in addition to talking about all of my IT issues, we were talking about the University of Minnesota. And so John has a fabulous collaboration with the University of Minnesota, and we were actually been able to get some animals from their unselected herd. And I'll get into that a little bit more. If you want to roll down one slide. For the sake of time, I'm going to blow through this real fast because y'all are all already in the dairy industry. You know that mastitis is really expensive. It's the most expensive cow disease for dairy cattle, estimated to cost $2 billion annually. And again, these estimates are close to 10 years old. And we know with the cost of everything now, not only are the inputs like antibiotics and labor going to be more expensive, but the cost of that lost production also going to be more expensive. We want to keep going. And then why we specifically were funded to look at mastitis mitigation is basically it's the biggest use of antibiotics in the dairy industry. And y'all all know that is expensive. And then what you have to do with withdrawal periods and all those kinds of things. So we want to go to the next slide. What you all seemed most excited to hear about was the old genetics. And so somebody actually said they had met Dr. Charles Young in the chat, and that was really cool. I would love to meet him because just the foresight and the things that we were able to do because of the decisions he made in 1964, incredible. So at that time, the University of Minnesota, and there's actually two other universities who did something similar at the time, but have since lost their herds and not followed through in the same way. He decided to establish basically a control population and said, okay, if we're going to start increasing this genetic selection for milk production, let's stop right here. Basically, we're keeping a living time capsule and say, hey, what is going to change? We will always have this to look back on. And so this herd was started with 20 breed average bulls for milk production in 1964. It's been a closed herd, although they did make sons of the original bulls sometime in the mid-90s. Sounds like there might be some people in this chat a little bit more familiar with them than I am. And that's who we're breeding to currently. It is a closed herd. Surprisingly, inbreeding is less than 6.25%. So again, when Dr. Charles Young designed this population, did a phenomenal job. And as you can imagine, the 305-day milk yield is about 4,500 kigs less than compared to contemporary Holstein. We want to roll to the next. And if you see right here, sorry, guy in the green shirt is my current collaborator, Dr. Brian Croker. And then guy in the blue collared shirt with the red cap is Dr. Charles Young that we were talking about. And this is one of the unselected Holsteins along with her calf. And you can kind of see, especially if you're used to contemporary Holstein cattle, I just have a totally different body style. Okay. So what has changed since the 1960s in milk production? So there's this beautiful slide here in orange and blue from the Council for Dairy Cattle Breeding. And you can see just what a tremendous change in milk production has occurred, not just due to management, but a good part to genetics. And what I want to shout out here is if you get around like that 2009, 2010 mark, you can see where this blue portion, the genetics portion, starts to get a lot bigger. And that's basically when genomic selection came on board. And so that's just really cool to see just the amount of progress we've been able to make with that genomic information. On the right, you can see actually two of my replacement heifers. And so they are a month apart in age. And actually the heifer with on the left with the orange tag is 1 month younger than the heifer on the right with the green tag. And so both born and raised on the campus in Ames, Iowa, same plane of nutrition, same management, very good plane of nutrition because these are government cows. And so the big difference here is their genetics. And so the gal with the orange tag would be a contemporary Holstein, gal with the green tag going to be one of our unselected gals. And you can see obviously the difference in height and then just the difference in body composition, for lack of a better word, they're beefier. And so you can see just looking visually, there's a huge difference in these animals. And so we're going to get a little bit more precise than that and talk about some of the genetics that have changed. So if we want to go to the next slide. So there's, again, been really tremendous work done looking at what are the genome changes. And so this paper that I'm citing here looked primarily looking at milk production, but also noticed, okay, since the 1960s, and especially since, 2009, 2010, we've also changed a large number of other genes. So you're probably pretty familiar with some of the fertility issues that have come up, and that's beginning to be addressed. What I'm going to talk about today is also the large change in immune genes. And so this paper reported, okay, not just a change in genes like DGAT1 that we know are really important for milk production, huge gene cluster differences in a lot of our important immune genes. So major histocompatibility complex, T cells, immunoglobulins, all the things that are really going to make an immune system functional. And so basically we're kind of trying to get through this question, okay, did it change because milk production and immune function are basically antagonistic and you can't have both, which may be part of the question, but we think it might be a little bit more of just, hey, we were only selecting for one thing, we weren't paying attention to anything else. We basically kind of bottlenecked down our population and kind of what we brought forward, we weren't paying attention to making sure we brought the immune system with us. Yep, that was perfect. You had perfect timing. So this is work that John Lapolis that I talked about did. And so this is what's really cool about working at the National Animal Disease Center is we can do studies like this. So this is an intramammary E. coli challenge. So basically grew up E. coli in the lab and stuck it up a teat of a cow to see what would happen. And so you can see in the blue here, you have those unselected Holsteins, and in red, you have contemporary Holsteins. And this is just looking at the number of those E. coli bacteria that were growing in the teat after challenge. So we know exactly how much E. coli was put in there in the first place. We can look at how that changed over time. And so you can see here on the bottom, we have basically just how many days after challenge there were. And so you can see in the blue, those unselected Holsteins, about 12 hours post-challenge, they saw a peak and then immediately came back down compared to contemporary. So anything that's got that asterisk is what we're going to say is statistically different. And you can see just a huge difference in the number of bacteria that were able to grow in these contemporary Holsteins. And then not really pictured on the slide, but also observed was just they had a greater systemic response. So basically the unselected cattle were basically able to localize that bacteria in the mammary gland, clear it, versus contemporary cattle got sick. And so really we want to know, okay, why are these unselected cattle so much more resistant to this E. coli challenge? If you want to go to the next slide. So basically these are the two questions that I'm trying to answer. Is what makes this 1964 herd so mastitis resistant? And how do we get it into modern cows? So real quick, I'm going to talk about the approach that I've taken. And we're going to talk about something called gene expression. So we talked about there are major differences in terms of what's actually in the genome in response to selection since the 1960s. We tend to think of genetics as something that's stagnant, like, okay, you've inherited it and that's it. What we don't necessarily tend to think about is actually genes are expressed all the time. So you cut your hand and your body starts to heal. All of that happens because genes are turned on or expressed. And so basically the biology in 5 minutes, the basics of everything is that DNA gets transcribed into RNA, which is basically instructions for how to make protein. And that's how your body or a cow's body is going to respond to any kind of challenge. So it's dark, your pupils dilate, gene expression. It's dark, you're looking out because, oh no, the cow's by the gate and your heart starts to race, gene expression. And so basically what I want to look at is, okay, we know the genetics are different. How do we kind of meet in the middle and say, okay, what genes are expressed in these different circumstances? We want to go to the next slide. So I started as a mastitis scientist in April. Before that, I was a postdoc in our brucellosis and tuberculosis unit, and was doing a lot of work with some different species there. And John came to me and said, hey, you want to work on this? Absolutely. And so kind of to get away from the question of, okay, we know milk production is really different between these two genetic lines, and that's going to complicate everything. Let's kind of start from the very basics and take the cow out of the picture for a minute. So what we did is we took 4 heifers from the contemporary line, 4 heifers from the unselected line, drew blood, and pulled out the immune cells that are circulating. So the PBMCs are just circulating immune cells, and we have them, cattle have them, your dog has them, everybody's got them. And we said, okay, we're gonna expose them to the vaccine strain of brucellosis just for 24 hours and see what happens. And so this has been a pretty well-studied system. And we're going to take all the RNA after 24 hours and compare, okay, what's different, and sequence it. We can go to the next slide. And so it was cool because we had huge differences in basically gene expression between these two genotypes. So you can see here we had 128 genes that had more expression in the contemporary Holsteins, shown that here in red, and 284 genes with higher expression in our unselected Holsteins, shown in green. And so I don't think y'all want to sit here and listen to me list off about 400 genes. So if we want to go to the next slide, I use some software to group them. And so basically what each of these big circles represents is just what those genes do. And so the larger the circle, the more genes were involved in that pathway. And so this is just the genes that were upregulated in our contemporary Holsteins. And we see here we have a bunch of these big red circles. They're called gene ontology terms. And they're all related to this alanine, aspartate, and glutamate metabolism. And if you're like, what is that? That's okay, because it's not something we talk about very often, because basically it's a stress response. And so what we are seeing in these contemporary Holsteins is that when we're looking at their immune cells, when they're challenged with the bacteria, the genes that they're expressing is they're just stressed. They were like me when I was trying to get this presentation to load. They're not getting anything done. They're just freaking out. And so if we go to the next slide, that is way different than our unselected Holsteins. So unselected Holsteins is what I was trying to be and have this presentation ready, right? Because they are just on the ball. So same thing, you notice right away, okay, hey, we've got a lot more of these big circles. We have a lot more different colors. And if we start to look at, okay, what are these terms that these genes were a part of, we get to see, hey, these are actual immune responses. So we have natural killer cell mediated cytotoxicity, really important. Natural killer cells are like Batmans of the immune system, like just swimming around, ready to eat something that's invading. Interferon gamma production, also really important because that's what's going to trigger a lot of your other immune cells to come help and say, hey, we've got this pathogen going on. We have T cell differentiation, regulation of interleukin 10 production. I mean, all of these things are characteristic hallmarks of an actual functional immune response. And so this is work that we just got published a couple weeks ago. And so really, really interesting. Okay, so we can see, yes, we've had these huge gene changes due to selection, and they have a huge impact on how these animals are able to respond even to just this little challenge of the cells in a petri dish. If we can go to the next slide. So kind of what we want to look at is, okay, so then the question comes up, are we seeing these differences because there's something going on with the different kinds of cells? And so I told you we took these immune cells out, we put them in a dish, we collected the total RNA. And so basically that's like looking at our bulk milk tank, right? Like we're getting an overview of everybody. It's not giving us a chance to look at any of those individual cells, which if you've ever had to suffer through an immunology course, there are a lot of them and they all have very specific jobs. And so what we did instead of looking at this bulk RNA is that we went to single cell RNA. And what that is basically putting an ear tag on each of these cells. So there's a company called 10x that's developed this system. It's called a gem chip. Basically, each of these immune cells that we isolated flows through one at a time, gets tagged with a little bit of sequence. And so we know and are able to differentiate each cell, just like we're going to put ear tags in our caps. I want to go to the next slide. So pretty much the same experimental design as the first, except we used 3 heifers instead of 4, because this is pretty expensive work. Isolated those PBMCs in the blood, and then instead of trying to pick a specific bacteria, so we know gram-negative bacteria tend to cause more severe mastitis because basically they've got this cell wall. We know that the cells are able to recognize this because it's basically got these little strings, LPS, this lipopolysaccharide structure hanging off. So we said, okay, we're going to try to get a big picture here. How do they respond to just seeing this thing that they know is a bacteria rather than trying to pick one specific bacteria? So we exposed these single cells to this LPS for just 2 hours. We want to go to the next slide. Then basically, I did a lot of computer work because even though I can't get slides to download, I can do some very serious coding analysis. And basically what this does is let me cluster all these cells together and figure out what kind of cells we do have. And so if we go to the next slide. So basically, in case you haven't noticed, I have a thing for dot plots. And so each of these dots in this case is a different immune cell. So in this case, on the left, we have the contemporary. And so in this case, each of these different colors is a different type of cell. So we've got really what we expected, right? All of the different kinds of immune cells. We've got B cells, we've got monocytes, we've got those natural killer cells, we have all the different T cells. What was interesting was that we had a couple that my algorithm couldn't figure out what they were. And so I'm still not sure if that's just because they are so different due to selection or because we had such low numbers compared to our unselected animals. So you can see basically from left to right, comparing contemporary to unselected, we have, for lack of a scientific term, way more dots in the unselected. That's because we have way more cells. And we started with the exact same amount of blood, followed the exact same protocol. And so it's really interesting that they have just so many more immune cells circulating in their blood. And so that right there says, okay, maybe this is why they have, is this why they have a better immune function? Is it because they just have more of these PBMCs? But I still didn't want to just stop there. I want to say, okay, what are the differences between the cells? And so this is current work that I'm working on. And so we can kind of see here these like pinkish red cells, B cells. in both are the most numerous cell. That's pretty normal. They should have 10 to 20% of these B cells. And so I said, okay, what are the gene expression differences between these? So if we want to go to the next slide. So what are B cells? I'm not going to give you guys an immunology lesson because I'm a trained geneticist, not an immunologist. We see our B cell here. Basically what he's going to do is point out, hey, there's a pathogen. And then B cells are not fighters. They're long distance guys. They're going to throw antibodies at it. But B cells are really important, not just for that first, basically, immune response, but also you're going to get later on what are called memory B cells. And we're all kind of familiar with antibodies, right? We want to make sure we have antibody production because that's how all of the vaccines that we use in our cattle work. And so really important for longer term because they're going to turn into memory B cells. And next time we see that pathogen, they're ready to go. So what was really interesting was just within just the B cells. And so if you are a statistics nerd, I did normalize this and divided, okay, what are our levels of gene expression divided by how many cells that we have. So we're not fairly biasing towards these unselected Holsteins that just have more cells. We did take that into account. And we saw a lot more genes with higher expression in our contemporary Holsteins. So 266 again in red and 377 genes with higher expression in our unselected Holsteins again in green. Again, don't think y'all want to hear me read several hundred genes. So kind of just try to get a first look and see what is going on with each of these. And it followed a very similar pattern to what we saw earlier. So we scroll to the next slide. This is what's going on as far as gene terms for these contemporary Holstein B cells. Literally nothing. So here we're again looking at these gene terms. We can have them either in red, molecular function, or biological processes. Basically, they're not activating a single biological process. And the two molecular functions that they are functioning are, again, just kind of this stress response. And this varies dramatically compared to when we look at the unselected Holstein B cells. So we had over 30 different molecular functions going on, and almost 600 different biological processes that were activated. And so I just tried to highlight some of the top ones, right? And so again, we have cytokine activity. So cytokines are really important. That's how your immune system, basically, those are their text messages. That's how they communicate with each other and say, hey, let's get organized and get going against this thing. Chemokines are just a specific kind of cytokine. And then again, we're seeing immune system processes, immune response, and then kind of what we expect B cells to do. They're saying, hey, other cells, get over here. And you see this granulocyte migration, leukocyte migration, neutrophil migration. And so this is ongoing work. So, hey, we've seen this huge difference in the B cells. Y'all saw just how many different types of cells that we have. And so I'm sitting through and going through each of these and saying, okay, what's different? And my hope is that there's going to be a couple genes that pop up that are differentially expressed that can kind of give us a chance to narrow down and say, okay, these genes are really important, kind of regardless of what kind of cells. And my hope is that we can either find something that we can genetically select for, which might be difficult, right, if it's already gone for modern Holsteins. As a geneticist, I'm always hoping for something that we can gene edit in, because then we're not just limited to Holsteins. I heard people talk about Guernseys or Ayrshires, but then you're getting into, you know, a really, which they might already have it, they might not need it. You're getting into a lot of regulation, gene editing still very highly regulated. Or, okay, so we talked about genes are expressing to make proteins. Is this going to be, okay, they're making a protein that is something that we could treat with as like a prophylactic. And so these are all kinds of the things that I am working towards. If we can go to the next slide. And so I sat here and told you guys, yeah, genes get transcribed into RNA, which makes proteins, and that's how biology works. Not always. So there's also epigenetics, so things that are not the sequence itself, but basically things that physically surround the sequence that can affect how it gets expressed. And so we have DNA methylation, which basically you can think of as the on and off switches of the genome. And so basically your DNA is made up of 4 base pairs, A, T, C, G. Specifically, methyl is going to bind to that C, and what it can do is kind of stop, basically turn off a gene, stop it from being expressed. And then also we kind of think about, okay, we are familiar with the double helix structure. We kind of know what a chromosome is. We don't always think about, okay, does this double helix get wrapped up into a chromosome? It gets wrapped around these proteins called histones, and kind of the way it's wrapped is kind of like trying to get your cow to the middle of the herd, right? Some things are not as accessible as others. And so this is other work that I'm working on is, okay, what is different, not just at the sequence level that we already know is super different. We already know expression is super different. I'm pretty sure some of these epigenetic marks are also gonna be really different. And so going forward, I'm also trying to follow up on these. Now that I have bored you to death with very serious genetics, we're gonna get back to the cow side. So we've talked about all the work I presented today was related to gram-negative bacteria, which for obvious reasons tend to be our more severe mastitis. Doesn't mean that's our only mastitis. We still also see a lot of gram-positive. So coming up in January, again, we've got another disease challenge, which is just awesome to be able to do. We're going to do strep uterus, which is gram-positive, and see if kind of we still see the same differences, or is this specific to gram-negative bacteria. We also had a stakeholder meeting led by DMI back in June, actually at NADC, which was really interesting. And what was kind of what I heard from that was that producers were less interested in kind of the traditional mastitis pathogens, like, okay, the strep, the staph, the E. coli. They were more interested in some new upcoming pathogens. And so one of the ones on that list was Klebsiella. We happen to have a Klebsiella strain that was isolated from a cow in Minnesota. So after we clean these gals up from our strep challenge, we're going to challenge them with Klebsiella. If you want to go to the next slide. That being said, I'm also taking input. So there was a lot of things like, hey, we want to see research in some of these, for lack of a better word, real spicy pathogens like Prototheca, which would be really interesting because I don't know how being an algae versus a bacteria would trigger some of these immune responses. Basically things like that that are kind of considered the cow killers. So if there is something that you say, hey, I'm a producer and I really want to see this looked at, let me know. Because right now I am the entire mastitis unit. So, and I am here to listen. With that, I'm also seeking advocacy. We want to scroll to the next slide. So we're facing the same challenges that commercial dairies are facing in terms of rising input and labor costs. And our budget hasn't changed in years. And so we've been underfunded, but we're kind of hitting this tipping point to where now my entire budget goes to keeping our dairy running. And I have no more money to do research. And so it's really frustrating because, again, I mentioned we're the only federal mastitis research unit. And now I am the only scientist. And we do not have money left now after paying for input costs to hire somebody else. And so if this is something that you think you care about, if you want to roll to the next slide, it's a very frustrating position to be in because I'm a federal employee. I'm not allowed to lobby Congress. So we are funded under this in-house appropriation right here. I'm not allowed to go say, hey, please increase our budget because I'm considered biased because that pays my paycheck. And so I have to sit here and ask for producers to say, hey, Congress, this is what matters to us. So if you would like to see this research continue, especially if you want to see some of the spicier pathogens, because that's going to be more expensive research, let your congressional reps know to up our budget. And if you want more information on how to do that, email me, email my big boss, Roxanne Matroni, who's our national program leader for animal health, or talk to our legislative analyst. And so basically, hey, thank you so much for attending today. Thank you for putting up with all of my technical difficulties. Better geneticist than I am presentation uploader. And then, yeah, let me know what matters to y'all, what you would like to see, and then if you would like to see us funded. And with that, I talked as fast as I can, Gail, to try to get y'all out of here. If the science thing doesn't work out, I'm going to be an auctioneer. And if anybody has questions, now's their time.
I should clarify, I have a hard stop at 1, but that doesn't mean that other people can't stick around.
Oh.
So I will just be disappearing, and so that's why I didn't want to have to share my screen, but I'm sure we're going to have a bunch of questions coming out after that talk, so I'll hand it back to Fred.
Okay. Thank you, Fred.
I would say we're a fairly small group. If anyone would like to just unmute and ask a question, that might just be the right way to do this.
So Kaitlyn, I know you're a geneticist, but would you anticipate, or are you going to look at other diseases? I think of other diseases that are nasty, like hairy heel warts, with those spirochetes in the foot. Would you anticipate the same sort of issue if you tried to look at that disease? Or
That is a
other,
great-
I'm just trying to think of other nasty infectious diseases we deal with.
Correct. And so kind of, it's like you're looking in my brain. So we actually have a spirochetes disease unit also. And so I've been talking with the scientist over there because she would really like to answer that same question. And so here's kind of, this is a finite population to begin with, and I do not have that many of these cows. And so what me and her are trying to plan is just some kind of digital dermatitis that she knows she can clear up. That way we can try this, okay, maybe in our replacement heifers while we're waiting for them to go into our lactating herd and be able to do some of our mastitis research. But yes, we would love to look at some of these other things. We have a finite amount of cows and money.
I have a question about I guess they call it a contemporary line. Have you looked at like picking out different lines within the contemporary group to say, hey, some of these genetics might be pretty good for immunity and some might not, and get more differentiation within that larger group?
That is a great question, and that's something that I'm currently working on. Because traditionally this group has leaned very heavily immunology, and I'm kind of the first geneticist that they've had. The issue that we run into is I can milk 20 cows at a time. So from a research design perspective, I want 10 of them to be unselected and 10 of them to be contemporary. And so I kind of run out of statistical power to ask any questions as to, okay, what's different between contemporary lines? And so if there's anybody on here at a university that wants to look at some of that with me, let me know.
Another question I've got, Kaitlyn, would you anticipate, or do you think there'd be a different vaccine response? I'm trying to think of if we could create disease, vaccines or something for this. So would you anticipate, I know these are a lot of speculative questions, but what's your suspicion on that?
So you're kind of loading a couple questions in there. And so I'm going to try to pick apart what I thought you asked, and then let me know if you were not heading that way. So the first thing you asked, is there a different vaccine response? And so I interpret that question is there a difference in response to vaccination between the contemporary and the unselected? I'm going to bet there probably is, based on those differences that we saw in B cells, which we know are the ones who are going to make antibodies. And so that's really nice, right? Because I have a built-in project right there. I have to vaccinate my replacement heifers every year. And so following up, I am going to say, hey, what does our titer response look like? And then you were kind of asking, okay, as far as us trying to make a prophylactic out of this, that is very pie in the sky right now because we have a lot of different things. But hey, it'd be really nice, like, right, traditionally, there's been a lot of really cool dairy research that's happened here in Ames. A lot of the stuff as far as like adding vitamin D to diets. And so hopefully we can find something out of these 1964s like that, where it's something we can get into standard management. But again, that's very speculative right now. And was that what you were trying to ask, or did I interpret that incorrectly?
No, that's fine.
Another question, with my animal health background, I like some diseases, the antibodies are really not what protects, it's really more the T cells. And like for BBD, for instance, so the titer levels don't really mean anything. It's almost the anti-protection. So are you looking at the T cell response too, or did I miss part of that? Okay.
So yes. So I have just started on the single cell data. It is a lot of information. And so far I've been able to look at the differences in the B cells, and then now we have all the different T cell subsets. And so I'm going to go through each of those individually. I just have not got to that point yet.
Okay, cool. Thanks.
I have a question. It's not really I know you can't answer this with your existing resources, but I'm curious what your thoughts are on it. We heard Dan and some others at the beginning kind of talk about, they're incorporating some older genetics into their herd. So what would happen if you took those 1964 cows and started selecting them, but started selecting them for traits like mastitis resistance? Like what would happen? What do you think there's options for some of these older genetics that people are incorporating in their herds?
That is a beautiful question. And so this is kind of my dream would be to make F1s between these 1964s and these contemporaries, and from there select a more balanced selection where we could have both milk production and mastitis resistance. Again, you're starting from a small population. How many generations would that take to make something that would be commercially viable? I don't know, but it would be really cool to look at.
I guess maybe my question too is this is way outside of your funding stream, but if we could get a time machine and go back to 1964, right? Because the idea is that those genetics are frozen in time. So it's kind of fun to think about. I guess this isn't a question per se, it's more of a thought. It's kind of fun to think about, like, back then, if we had different selection criteria, what would we be investigating right now? And what would our cows today be looking like?
For
It's kind of
sure.
just a fun thought experiment.
Is it possible to use gene splicing to pull those desirable genes and actually put them directly into modern cows and avoid XXX generations of trying to get the milk production up in them?
Hi, Dale. Thank you for that question. That would be my dream. So there are two things that make that difficult. One, regulation, and then two, which hopefully we will be able to overcome, is that most things are controlled by more than one gene. And the immune system, as varied as it is, probably more than one gene we've got going on. And so we hope we can identify one gene with enough effect that we can then edit and splice that into modern cows. That's the goal as far as trying to map all this out. I hopefully in the next couple of years we'll have an answer to that question.
Go ahead.
Yeah, Kaitlyn, just a quick question here. So in any of the work that you've done, there are two things that are possible with selection, of course. One of those is, is the impact that you're seeing on the immune response related to correlated response to selection for other traits, or is it simply because of the increasing inbreeding in that is brought about by selecting for the same traits over and over again?
That is a great question. And so I actually just saw a talk by John Cole, who's a very well-known geneticist, and it was really interesting because he was talking about the effect of inbreeding in dairy cattle. And it was basically discussing the trade-off between, as we're selecting, okay, we're improving the trait we're selecting for, but at the same time, then we're also creating inbreeding. And basically, milk production, we're able to improve through selection more than we're going to be able to harm it through inbreeding to this point. And that was true for a lot of the current selection traits. The one trait that wasn't really true for was when they were looking at somatic cell count. And so that is where I think this is a very interesting trait to look at is, okay, when we are looking at selection, the inbreeding is causing, even if we are, how many producers and what are the value that they're putting on that SEC score? But the basically inbreeding trade-off that comes with that selection is harming mastitis resistance quicker than it's improving it, which I thought was pretty interesting. And then there's the first part to your question that I'm afraid I've forgotten.
No, I think you hit it. Yep.
Okay.
Yep.
Early on in the presentation, you'd mentioned that you saw differences in breeds. And my question then is that potentially a consideration? I know well over double-digit percentage of the herds here in Western Iowa are crossbreeding. Should we be looking at some different bulls? I mean, go ahead.
Oh, sorry. So I told y'all, right? I come from a beef background. A lot of my research previously has been in crossbreeding beef. So that's something I would love to look at. If anybody would like to look at that with me. I don't have enough animals to really crossbreed here, but if somebody has a population they'd like to study, let me know. And then can I ask, when you're saying crossbred, are you talking with Jersey, Guernsey? What do you mean by crossbred?
At this point, I'm looking at, you know, the standard rotation, a Holstein, Dane of some kind, Jersey. That's kind of the pro-cross matcha, and I think we're seeing more herds of that. But, you know, if I knew that I could reduce incidence of mastitis and lower my cell count by using Illawarra, you know, I'm certain we would see that come about. Dale, did you catch my plug for milking shortened horn genetics there? I just wanted you to be aware.
Came out so loud and clear, I may have to leave.
Are you looking at any, well, there's really no old breeds. I'm thinking of Jerseys if they've changed, you know, they're the second most popular breed, but there's really no old genetics. There's probably maybe some semen or something around. Like Dan said, he's using really old Guernsey bulls. So you could theoretically look at something like that. I think this whole thing is really interesting. What breeds have we bred out? What genes have we bred out of our animals that might be really valuable?
Thank you. Yes, I'm sure, because I worked for Bull Stud for a little bit, and we still had glass ampules of semen from that long ago, as before they even had straws. So probably somewhere out there has it. I'm not aware of a population of Jerseys like what we have with these unselected Holsteins.
It's always amazing when you start digging around in a retired dairyman's jug, what you're gonna find there.
Kaitlyn, there's some work being done at Penn State with old genetics. Dr. DeChaud, I think is his name. I'm not quite sure. He writes in Horge Dairyman all the time. Have you had any contact with that set of genetics at all?
I have not. Thanks for bringing that to my attention. I will follow that up. Could you drop that DeChaud name in the chat? I'm not quite sure how you spell it.
Yeah, it's so that's Chad Deckow. And
Okay.
we actually hired as a faculty member the graduate student who was working on that. So Isaac Hagan is now in a dairy extension position here at the University of Minnesota. So would be a good collaborator on this, and you know, definitely has an interest in those old in the old genetics. Most of what they did were bulls from this 1964 control line, so it was really the same set of bulls. But a lot of that was accessed through the USDA animal germplasm preservation program out in Colorado Springs. And Dr. Deckow actually did a postdoc out with that group as well. So before he
Okay.
came to Minnesota.
Thank you very much. I will reach out to him.
We have had a really great discussion. We can go a little bit longer if somebody wants to insert another question. We'll keep the discussion going for a few minutes. If not, I like to thank everybody for sticking with us. We know we had some tense moments there, but we got it figured out. Remind everybody that there in the chat box I have both the evaluation and this will be archived at the Iowa State Dairy website, as well as the I-29 Moo University website. Give us about a week to put it together, and then I will send out a notice that it is posted and ready for viewing. Kaitlyn, thank you very much for bringing your research to life for us. We appreciate the information, and hopefully as folks go through the end of the year and say, hey, I'd like to see that research move forward. So maybe we can put a shameless promotion in for some research dollars.
Thank you very much.
Thank you, folks. This will conclude the program.